ये बंद हो गया. So we have 3 series of lectures. When we talk about vectors in human health, vectors means, please come this side. इधर आके बैठो आप लोग सारे बीच में. What do you understand by vector and what is a pathological organism? 123. क्या vectors are? So vectors are the one which cause the disease or is something else? They just transmit the disease. The disease causing organism is something else. That is what we are known as vectors. Sit down. And a large proportion of diseases in human health are caused by vectors, and that is why it is essential to study that as a separate entity, that what are those vectors and how, what diseases they cause and how that can be controlled to, how that can be used to control these diseases. So the outline is, there are 3 series of lectures when we talk about vectors in human health. The first one is today, that is epidemiology and health effects. The next class would be on surveillance and management of precisely these vectors and these vector-borne diseases. Individual diseases will be taught as a separate lectures in the epidemiology of diseases that you are talking about. This is essentially dealing only with vectors. Please come and sit on that side. That side. And finally is the programs and policies of environmental health. Today's lecture, apart from this, is epidemiology of common vector-borne diseases. What are the vectors of medical importance? Because there are many insects, there are many arthropods. You have read in your 12th standard science class. They are arachnids, they are arthropods, they are crustaceans. But we don't need to know each one of them. We are obviously dealing only the ones that are important for medical this thing. And what are the characteristics important for control? What characteristics you should know about which are important for controlling these diseases. So amongst the risk, please close the door behind you if there is no one else. So about 80% of the world's population is at risk of one or more vector-borne diseases. So each one of us is at risk of developing any one of the vector-borne diseases. How many of you had malaria from your childhood up till now? Malaria किसी को नहीं हुआ? डेंगी? डेंगी हुआ है? कहाँ हुआ है? हॉस्टल में? So how many of you had malaria? Nobody in your childhood got tested for malaria? Dengue? No one? Chalo, good. Then you are not in that 80%. The burden is 70% of the global burden of communicable diseases is due to vector-borne diseases. So in terms of percentage, about more than 10%, which is a large numbers of diseases are caused by vector-borne. And it is a known fact that more people have been killed by mosquitoes than by all the wars in the history that have happened. So that's a very important fact, and that is the importance in terms of burden of the disease of mosquitoes. So mosquitoes are what should I say, biologically they are more evolved even than humans, and they are evolving up till now, which are posing challenges for many of these vector-borne diseases that are there. And about 700,000 deaths are caused by vector-borne diseases annually across the globe. One of the 2 most important factors when you talk about vector-borne diseases is, one is climate change, which is causing a lot of change in the environment that is there, which is causing a lot of change in the vectors, their spread. And ultimately, because of this interstate and inter-country travel, you have vectors moving from one country to the other and otherwise also. Okay, and rapid unplanned urbanization. These vectors are now having cycles even spilling onto winter months. So earlier we used to have malaria and dengue only in the summer months or in the rainy season, or maybe up till October or this may considering tropical climate. But because these vectors are now learning to survive and ecologically they are more evolved, we also have cases of malaria, dengue, other vector-borne diseases even splitting into January, February, or even up till December. So the surveillance has to go on across the across all the 12 months to understand which diseases are occurring. So it is not that if you are getting a case of fever with malaria in the month of November, you can entirely rule out dengue. You will have to maybe talk about fever with a rash. You have to keep dengue as your differential diagnosis, and that is why you need to understand this. So, the modes of disease transmission, when we are talking about vector-borne diseases, there are about 5 modes. One is direct contact, that is person to person transfer. When two hosts are in direct contact with the other, the arthropod vector itself gets transferred from one host to the other, that is pediculosis and scabies. That is vector mechanically transports from one person to the other. You are not allowed, please leave. The vector itself transmits, jumps from mechanically from one person to the other, and the examples are pediculosis and scabies. The second is mechanical transmission, where they work as carriers. The disease causing organism is transmitted to the outside or inside of the bodies of the arthropods without undergoing any development, propagation or any changes in the pathogenicity. These are diseases transmitted by houseflies such as diarrhea, dysentery, cholera, hepatitis A and E. So, just because disease causing organism, these bacteria are sticking on to the proboscis, the feet of these houseflies, that is how this mechanical transmission happens. Now, you need to know these mechanical transmission because since you are so absent in your classes, we have seen your seniors writing down as malaria being transmitted by houseflies. How is malaria transmitted? Anyone? Which mosquito? Anopheles. So, please be aware and please open your eyes and ears when you are listening to these classes, because if you are telling that malaria is transmitted by housefly, that is very, very shameful for you also. Okay, so don't do that. Please learn that housefly is transmitted most of the diseases only by mechanical transmission, acting as carriers. They do not, the organism does not go any developmental stage inside the housefly. Then we have the biological transmission, which is the definition of vectors. The disease causing organism undergoes certain biological changes inside the body of the vector. Depending on the type of biological changes, the biological transmission has been further classified as cyclodevelopmental, cyclopropagative and propagative. Vector-borne diseases, these are very, I think 4 to 5 MCQs do come from this particular section. So, it is a factually laden chapter as such, so but you should be knowing these facts, that which are the organisms or vectors which are having a cyclodevelopmental cycle, a cyclopropagative and a propagative. So, cyclodevelopmental is the part of the cycle and growth happens, but no multiplication, that is Wuchereria bancrofti, which causes filariasis. Okay. So, Wuchereria bancrofti has a cyclodevelopmental cycle, where inside the humans and inside the mosquitoes, it goes about change in the development, but not a change in the number of the organisms that are transmitted from the humans to the insects. So, that is a cyclodevelopmental cycle. Then we have the cyclopropagative cycle, where a part of the cycle also happens, the growth also happens and multiplication also happens, and the common example is Plasmodium and Leishmania, which causes Kala Azar. Please, you are not allowed, you are late. Then we have the propagative cycle, where no part of the cycle, only growth and multiplication, example Yersinia pestis. All arthropod-borne bacterial, viral or spirochetal diseases are the ones where we have the propagative, that is no part of the cycle happens, only multiplication, that is Yersinia pestis, which causes plague. So, you need to remember what are the diseases which have the cyclodevelopmental, which are the diseases which have the cyclopropagative, and which are the diseases which have a propagative cycle. So, this is the cyclodevelopmental cycle of Wuchereria bancrofti, where you have this mosquito takes a blood meal, that is L3 larvae enter the skin, and once these larvae enter the skin in the human stages, it develops into an adult. The adult produce sheeted microfilariae that migrate into lymphatic and the peripheral blood circulation. You must have read this in micro already. This cycle must have been taught to you in micro. So, once that is done, the mosquito takes a blood meal, ingests this microfilariae. Microfilariae sheds the sheaths and penetrate to the mosquito midgut and mitigate to the thoracic muscles. Then the L1 larvae, the L3 larvae, and ultimately migration to the mosquito head and proboscis. And when it goes on to to bite the another human, again this thing happens. Okay, so this is what is the cyclodevelopmental cycle. You have just the two male and female that develops, it do not increase in number, it is only inside the mosquito where the microfilariae number increases. Then this is the cyclo-developmental cycle of Plasmodium. Plasmodium cycle you have been learning since when? Malaria ki cycle since when you have been reading in school? 8th standard, 9th, 10th, 12th. So this complicated cycle should be known to you by heart, and you should know that this is a cyclo-developmental cycle, increase in the number as well as the cycle. So we have the asexual and the sexual cycle happening in the humans and the mosquito. So that's the cyclo-developmental cycle of Plasmodium, wherein you have this mosquito takes a blood meal, injects the sporozoid, they go to the liver, we have the exoerythrolytic cycle, then we have the erythrolytic cycle, then ultimately you have these falciparum gametocytes. The mosquito takes a blood seal and ingests the gametocytes. The gametocytes have the sexual cycle inside the mosquito, and ultimately the mosquito becomes infective to bite other human and takes place. This cycle is of how many days? After how many days does a mosquito becomes infective after intaking the Plasmodium parasite? Anyone, if you remember? This has relevance, why? Because the earlier you kill the mosquitoes, the earlier it is an important measure to control the transmission of the Plasmodium parasite through mosquitoes. So the duration of this cycle is important for control of diseases at the public health level. Okay. Then the modes of transmission, apart from this, we have the disease causing organism undergo certain biological changes, this we have read. Then we have the trans-ovarian and the trans-stadial cycle. The trans-ovarian is a disease organism is transmitted to the eggs through the ovary of the infected female, that is Orentia sushogumashi, the agent of scrub typhus transmitted by thromboculate mite, that is Leptosporidium deliense. It's a very common MCQ question, that trans-ovarian cycle is by the scrub typhus. Then we have the trans-stadial, the disease causing organism is transmitted from one stage to the another, that is tick typhus organism, that is the Rickettsia conurae, transmitted for infected larva to the lymph to the adult. Again a very important question, and even though we do not read a lot about tick typhus or scrub typhus, these are important diseases when we talk about public health importance, because of climate change and because of urbanization we are encroaching upon the forests and the other plantation areas. So that is why these diseases are pretty common, and the burden is really not known because they are not kept as a part of differential diagnosis very commonly, but diseases are quite common. So coming on to what is the I've just taken these slides only of malaria and dengue, but all of these diseases are equally important when we go to these further slides. So when we talk about malaria, you have to think about maybe Africa, and fortunately not much for India, because globally in 2033 the number of malaria cases were estimated to be 263 million with an incidence of 60.4 cases per thousand population at risk. There is an increase of 11 million cases from the previous year, and when you talk about malaria, you have to think about Africa, because 93% of the burden is borne by the African countries, and in African countries we have Nigeria, Congo, Uganda, Ethiopia. India is somewhere over here. Where is it? Yeah, here. So we have in terms of burden, a very small proportion, but since we have a large population, we do deal with a lot of malaria cases. But when we talk about the stages of malaria elimination, India is nearing the stage of malaria elimination and control. Okay. Then we talk about dengue, but dengue remains a very a tractable problem when we talk about even in India and across, because malaria also has subclinical cases, dengue has more subclinical cases and more of cases which are of the milder form as compared to deaths. So a lot of cases go unnoticed, and that is why, but we have to know that these particular belts in the tropical areas are the ones which are known to have endemicity for dengue, that they have these dengue outbreaks continuously. We have large outbreaks every 3 to 4 years can be expected, and there has been an increase in the number of dengue cases across these countries, even in South Americas, in these Mexico cities, in India, China, even in Australia, some of the African countries, and these countries which are a part of the Southeast Asia. Okay. So, it's dengue and malaria are largely tropical diseases, but they are also now moving on to the temperate climate because of the because of the climate change. So, insects of medical importance is you need to know about the mosquitoes, sandfly, houseflies, fleas, louses, tick, mites, cyclops, red-ovate bugs and tsetse flies. So, these are the ones, the names that you need to remember and what are the diseases that they are causing. Please stop talking. Anyone who is talking will be asked to leave the class. If you are not willing to listen, do not disturb. So, these are the mosquito-borne diseases. You have to remember this, there is no other option. You can make mnemonics, any shortcuts, whatever is remembers, but you should be remembering that these are the diseases that are caused by these vectors. Malaria by Anopheles, by Plasmodium species, and the reservoir is only humans. Humans are the only reservoir when we talk about malaria. For some of these species, other reservoirs are also known, such as catarrh, etc., which are the anthrophilic species, but by and large the reservoir for malaria is humans also, and that is how this transmission cycle is sustained. When we talk about filaria, the vector is Culex, which is very commonly seen, Aedes nivis and Mansonite species, but for our importance you should remember that filaria is caused by Culex quinquefasciatus, that is the vector for filariasis. You should remember that malaria is caused by the vector of Anopheles. And then the causal organism is Wuchereria bancrofti and Wuchereria that is nocturnal, and Wuchereria bruguiamalae, which is diurnal suppurative, and this is the, along with the vector, the causal organism might also change. But if you remember these words, that filaria is caused by Culex and the causal organisms is bancrofti, that is good enough for us, and the other one is bruguiamalae. So, these are the 5 things that you need to remember. The reservoir for even for filaria is largely human. So, the control has to be focused only on humans and the vector and the elimination of this causal organism. Then we have chikungunya caused by the Aedes species. It is an arbovirus. A virus is the causal organism, which is a viral disease, and the reservoir is also the humans. Chikungunya does not cause a lot of mortality, but it causes a lot of disability because of the severe pain in the joints and the body, which happens as a part of chikungunya, and that can last even up till years for many of the patients. Then we have dengue fever and dengue hemorrhagic fever, again by Aedes, the arbovirus group virus, the reservoir are humans. Then we have the yellow fever, which is also caused by Aedes, by an arbovirus group B virus, and the reservoirs are humans and monkeys. What you need to remember is about yellow fever is that even though we have Aedes species, which is a very efficient vector of this yellow fever virus, we do not have yellow fever cases in India because we do not have the arbovirus, and because there is a very strict control about the vaccination that is given for yellow fever. Do you know that the yellow fever vaccine is given at selected places across India, and AIIMS Bhopal is the one in central India. Okay, for all of Madhya Pradesh, anybody who is travelling outside to the countries where yellow fever has been seen, they have to take a vaccine of yellow fever from AIIMS Bhopal, get a certificate, then only they are allowed to get a get a ticket and travel to these countries and come back. Okay. So, even though we have the Aedes species, that is this particular mosquito is there, it causes dengue, it causes chikungunya, but we do not have yellow fever, even we do have the vector of this yellow fever causing vector, which is present largely in India. It is very commonly seen. And then we have, but we do have Japanese encephalitis, which is again caused by Culex vishnui group. This is filariasis and JE, both by Culex. This one is by Culex quinquefasciatus, and this is by the Culex vishnui group. Again, it is a viral disease, and mammals and birds are also the reservoir, and the cycle of Japanese encephalitis is quite common. You have mammals such as pigs, swine birds, which are reservoirs and which are important in transmission of Japanese encephalitis. When the individual diseases will be taught, then you will have to know about that. But this particular table is something that you need to remember, learn, and learn it by heart. Okay. Secondly, the sandfly borne diseases. You need to know about leishmania or visceral Kala Azar. It is seen in certain states in the eastern part of the country, that is Bihar and Chhattisgarh. The vector is Phlebotomus argentypus and the causal organism is Leishmania donovani. The reservoir is again humans and certain mammals also. For the oriental sore, the vector is Phlebotomus papatsi. The causal organism is Leishmaniae tropica and humans. Then these are probably the ones that you need not remember, but these are the other sandfly bone disease such as espundia and sandfly fever. But what you need to remember and not need to forget at all is that sandfly causes is a vector for Leishmania and visceral leishmaniasis and cutaneous leishmaniasis. Although diseases are some of the diseases such as dengue, malaria, leishmania, river blindness, they are counted under the category of neglected tropical diseases. Means they are tropical diseases. They were causing a lot of deaths and morbidity earlier, but for many years they have not been into the main focus, but they continue to be an important public health problem. So they have been categorized or clubbed together as neglected tropical diseases, and Kala Azar, dengue, malaria are one of not malaria, dengue, chikungunya, river blindness. These are some of the diseases which are known as NTDs. Okay. Then we have these housefly borne diseases. I just hope that you do not get confused that these diseases are caused by mechanical transmission by housefly, and they have bacillar dysentery, amoebic dysentery, gastroenteritis, typhoid, paratyphoid, cholera, poliomyelitis, viral hepatitis, trachoma and yaws. These are all the vector is Musca domestica, and the causal organisms are mostly the bacteria, Shigella, entamoeba, and even some parasites that are there, and reservoir are mostly humans. So housefly is a very efficient transmitter of many of the diseases, and fecal-oral transmission is the one which happens for some of these diseases. So this you should remember. If I asked what a vector is a vector for which of the diseases, you should be able to tell us. Then we have flea-borne diseases, where we have plague, which is the bubonic plague caused by the Xenopsella species. The causal organism is Yersinia pestis, which is what kind of cycle? Developmental, propagative or cyclodevelopmental? अभी तो बताया है. Last person, stand up. Yersinia pestis, what kind of cycle of biological transmission? Next. Next. आगे आगे नेक्स्ट. कौन सी साइड से खड़े होंगे? हाँ, दिस साइड से, फ्रॉम द लेफ्ट. Yersinia pestis, what kind of cycle? Next. Next. The entire class is going to stand up. अब तो गूगल करके ही देख लेते. Anyone? बोलो बोलो. Propagative cycle. Please sit down. And the reservoir are rodents. Then we have the endemic or the murine cycle by the Xenopsella species. The causal organism is Rickettsia typhi, and again it is caused by the rodents. Then we have shigerosis, diphyllidium, hymenoplosis, and these are the other diseases which are caused by flea-borne, but these are the ones that you need to remember precisely. Okay. Then we have Loud's bone. All of this you have to remember. For some reason, we do not remember that this epidemic typhus is caused by pediculus humanus, but these constitute a very important MCQ question. It is very, very commonly asked. So there is no other option but to remember that epidemic typhus is caused by Rickettsia prowazekii. The vector is pediculus humanus, which is very commonly seen, and reservoir are the humans. The epidemic fever or the relapsing fever, it is known as, that is caused by Borrelia recurrentis, and again the vector is the same, that is pediculus humanus, and the reservoir are again humans. Trench fever, it is a Bartonella quintana is the causal organism. The pediculus humanus again is the vector for this particular disease, and the humans or the animals are again reservoir. And dermatitis, which we see commonly is pediculus humanus capitis, Other organisms might be there and humans are the reservoir. Okay. So, dermatitis the secondary organisms, but the vector is pediculus humanus. So, this is the table that you need to remember for louse bone diseases. Tick bone, again hard tick and soft tick. Hard tick causes 3 diseases: Carcinur forest disease, tick typhus and tularemia. Soft tick causes relapsing fever. Hard tick KFD is caused by a viral disease, tick typhus is caused by Rickettsia cannori, tularemia is caused by Pediculus tularensis. Relapsing fever of borreliosis is caused by Borrelia duttonii, and these are the reservoir for all of these diseases. There is no other way to simplify it for you, to make it interesting for you. You have to remember these disease causing organisms, their vectors and the disease that they are causing. Okay. This Crimean, these are diseases which were actually not a lot was known about the diseases. Now with the advent of climate change and maybe of better diagnostics, we are now even hearing about these diseases such as Crimean Congo hemorrhagic fever transmitted by ticks and wild domestic animals and asymptomatic carriers. So we have the mite borne diseases. So we have louse, ticks and mites. Mites may again you have scrub typhus. The vector is Eldeliense and the causal organism is Orentia shushugumashi. Then we have rickettsial pox by Rickettsia acari, and then you have scabies. Now even cyclose transmitted diseases such as dracunliasis and fish tapeworm by cyclops species is something that you should know, but these are not very common and obviously they are not very diagnosable diseases because they are very, very rare. Then redovid bugs, Chagas disease caused by the redovid bug and the causal organisms Trypanosoma cruzi, and they are the animals are domestic or the humans. Then we have the tsetse fly, something has happened with the this thing in this particular system. Then the disease is trypanosomiasis, the causal organism, and the vector is Glossina species. The reservoir is wild animals, cattle and humans. So why you need to know more about mosquitoes? And you need to understand that there are basically only Anopheles, Culex, Aedes and Mansonia are of importance in India, and these are the ones that you will come across in various diseases of public health importance also. They are classified as Anopheles, which comprises only one important genera, that is Anopheles, and Culicines, which is comprising 3 important genera, that is Culex, Aedes and Mansonia. For all of these diseases, only the female are the parasites. The gonotropic cycle is of 48 hours, that is period from the blood meal to the laying of eggs. And they are attracted by body odors or carbon dioxide and heat, and males can be identified by the antennae which are more bushy. So whenever you are in a park and you see a lot of males, lot of mosquitoes on your head, they are most likely males which are there to infect or have the for the copulation with the female which might be biting you at that particular point in time. So that is why you have this big this thing over your head, which is because they are attracted by body odors. All important vector species are preferentially anthrophilic, that is they are mostly attracted to humans. जो भी आपकी disease causing है obviously, and that is why they are causing diseases in humans. The time of feeding is also variable. Some species prefer dusk for their blood meals, others late at night, while still others select early morning. This particular thing is important for control. So you should be knowing that which particular vector has what preference of timing of the meals that they are taking. These are important for control. The knowledge of habits is useful for designing control measures. So this is a mosquito. It has a head, a thorax and an abdomen. The life history, you have an egg, then we have the larva, then we have the pupa, the adult emerges, and that is how the life cycle completes, and that is about 48 hours for most of these mosquitoes. From an entomological point of view, it's there are specialists who study mosquitoes, their eggs, their pupa, their larva, but you should be knowing it for for your control measures that Anopheles, Aedes and Culex, the eggs are laid singly, they are boat shaped and they have lateral forts. Aedes have single eggs, cigar shaped without any lateral forts, and Culex lay their eggs in cluster or rout which are long ovoid shaped and they do not have any lateral floats. What are the larvae? They are free swimming creatures that feed on the algae, bacteria, and they themselves might be a food for some of the certain fishes, and that is why fishes are used for control of these larvae. They are 4 stages of growth, you need not know, but you can be asked about what are the kind of larvae for each of these species. So, Anopheles larvae, they float horizontally on the surface of the water. Culex have head downwards and Aedes also have head downwards. The difference is they have the Anopheles do not have a siphon tube which is used for breathing of these larvae, and Culex and Aedes, they have a siphon tube, but it is long and short. Okay. Anopheles are very active, Culex are slow, snail like, and they are slower. So, when you go in your CPSCR class or in FAHAP posting, you are asked to look for mosquito breeding. If possible, you might be able to even detect whether this particular family is at risk of malaria or of dengue depending upon, or of filariasis depending upon the vector that you are seeing. The pupa are comma shaped, but they are motile, they do not have feeding, they are the resting phase of the life. It lasts about 1 to 2 days and the adult emerges from the T-shaped slit. Adults, the Anopheles sits at an angle except for Anopheles facies. Then this is how you differentiate between Anopheles and Aedes. Most of you must have seen Aedes. It is known as the tiger mosquito, which is black and white in color, and now in the summer months they can be a very important this thing of fever cases that might happen in your hostel, because it is seen in artificial water containers. Then they have loud mouth parts, short palpi, and these things. The important thing that you to remember is Anopheles breeds in clean natural water, Culex breeds in dirty stagnant water, and Aedes breeds in artificial collections such as coolers, tires, or other flowering pots. These are the preferences for. So, you have to know these things, that which one breeds in clean water, which one breeds in dirty stagnant water, and which one breeds in Aedes. So, you will have in and around this campus, you will see all these 3 kinds of mosquitoes breeding, maybe not Anopheles, but definitely Culex and Aedes. Then depending upon the breeding habits, the various species of Anopheles are there, like slow moving water seepages or terrace rice fields, Anopheles fluviatilis, brackish waters, Anopheles sundiacus, well cisterns and overhead dranks, Anopheles stephensii, tank pools, burrow pits and ditches, and forest pools, streams and slit trenches by Anopheles dirus. Now, why are these important for us to know is because these breeding habits, such as these specific habitats, wells, tanks, brackish water or forest pools, tell us about what are the key where these particular vectors are going to be found, and these are important for control. So, when we do a vector surveillance, which will come in your further classes, we specifically look at these sites to determine that probably this particular population has a larger propensity if it is surrounded by more well, tanks or brackish water. You have to identify the breeding sites which help in targeted larval control. Then there is also vectorial capacity, which is calculated by a formula, and ultimately the value that we get is that if this vectorial capacity is more than 1 or by less than 1, it means an epidemic is likely. So, when we talk about vector-borne diseases, study of this vector biology is important to prevent imminent epidemics of outbreaks of various diseases, such as we very commonly have these dengue outbreaks occurring in your hostels. Okay. So, there are certain indices which will determine the kind of larva that is there, will determine whether a dengue outbreak is likely to occur or not. So, we do a kind of vector surveillance that is done there. So, we do it even for this thing, and the vectoral capacity is the density of this thing. So, some of these insects, even in less numbers, they might cause disease, and some of these insects or mosquitoes in large numbers, they might cause disease. So, Anopheles philippensis is transmit even at lower densities, but Anopheles facies needs higher density, and also Anopheles dirus. Then feeding practices, most of them are anthrophilic versus zoophilic behavior determines the control strategies, that whether you need to focus on animals being kept away from, because in the rural areas you have animals living in sheds which are close to the houses. So, if we have a species which has a zoophilic behavior, to control these or to prevent the transmission, you have to have these animals staying away from the houses so that they do not come inside the houses and bite you and you get the disease. Biting time determines the timing for interventions like bed nets or repellents. Now, further from these slides, we have a lot of MCQs where you will be asked. So, anybody who is not listening will not get an attendance. Okay? Although they are very simple MCQs. Then we have the resting behavior. We have the endophilic mosquitoes are more susceptible to indoor residual sparing. So, in the next class, when you talk about how these diseases are controlled, you might have seen that a vehicle which is letting out smoke to kill the mosquitoes. It is seen there even in your hostels. And now during the summer months, you have that person that comes to your individual rooms and does the spraying. What is that? Spray करता है ना आपके यहाँ पे नीचे on the walls in your rooms. What is that? Did you have it done in your hostel? You have been there in the college for I think third semester हो ना, third year. हुआ है कभी आपके hostel में? Have you seen it? So did you never wondered what is this person spraying and what is he doing? That is known as indoor residual spraying. They put an insecticide on the wall so that when the mosquito sits on that insecticide, that mosquito dies. Okay, and when you do have fogging, wherein you have machines or vehicles which blow out this smoke, which is again having the killing properties for these mosquitoes, they are killing the mosquitoes while they are flying. So we have to know that which mosquitoes are likely to sit inside and which mosquitoes are likely to be killed while they are outside, because that is the preference of mosquitoes. When you have these Aedes mosquitoes, they are likely to come and sit inside the in the walls or in the curtains. So that is why we ask these people to even put the insecticide on the curtains, and that is what is known as indoor residual sparing. And once it is done, it lasts for about 2 to 3 months. It will come in the further classes. And then we have the flight range. It has defined the radius of control efforts around the breeding hotspots. So Anopheles diarrhea has about a radius about 1 kilometer. Culucifacius is about 2 kilometer, and Anopheles sundiacus up to 8 to 10 kilometers. So the fogging that is done, if we have a case of malaria and we want to understand for how many houses or for how many kilometers we have to do the fogging, is this if the vector can fly up till 10 kilometers, and that is how you need to know about the flight range. Longevity, longer survival increases transmission, influence the use of long lasting interventions. The mosquito should live at least for 10 days post meal for plasmodium development. Insecticide resistance, it's a very important and crucial for choosing effective insecticides and rotating the chemicals, because this is a very big problem when we talk about malaria control, especially in the African countries and even in India. Insecticide resistance to many of these for many of these vectors is a very big problem. So coming to your MCQs, let me stop the screening first.